is October 30th, 2013, and today Dan Orr is going to lead us through a very interesting talk on computer simulations of protein folding. And it's of particular relevance because of the recent Nobel Prize awards given out in this field. So I'll let Dan introduce himself a little bit and tell us a little bit about his areas of expertise and lead us on the protein folding discussion.
Okay. Thanks, Nina. Can everybody hear me okay?
Yeah.
Okay, great. Let me see if I can, I'm going to try the screen sharing thing. And, So my name's Dan Orr. I'm a lapsed GenSpace member. I'm a patent attorney, and I continue to serve as GenSpace's pro bono counsel. I'm also a recent graduate in molecular biology, and starting next semester, I'll be starting a master's program in bioinformatics, as well as next month, I'll be starting a job with the FDA in the Office of Drug Evaluation Research. That is assuming I pass security clearance. So I wanted to, this article has particularly interested me, not only because of the recent Nobel Prize, but also because, from as far as I can tell, my relatively small knowledge in the field so far, this is really one of the founding articles of the field of bioinformatics. I hope that since this article is almost 40 years old now at this point, that we can kind of use it as a springboard, not only to talk about what has been done in bioinformatics and how the field has come about, but also what's left to be done in the future and the more general role of computer simulations and in silico experimentation in the development of molecular biology and biotechnology. So just briefly, the kind of four basic categories of structure that we talk about in proteins are, of course, the primary structure, the amino acid sequences. The secondary structure are the more general categories of shapes, the helices and the beta sheets and the loops. Tertiary structures when we start getting into 3 dimensions, and then the 3 dimensions of individual amino acid chains, and then the quaternary structures when we're talking about a protein that has multiple amino acid chains, the relationships among those chains within 3-dimensional space. So two of the article that we're talking about this evening, both authors were 2 of the 3 who won the Nobel Prize in chemistry this year for the development of multi-scale models for complex chemical systems. Just a little bit about their background and how their research came about. I found it helpful in understanding the article, or more precisely why there was so much in the article that I didn't necessarily understand. Levitt and Warshel originally met at the Weizmann Institute in Israel in 1967. Both were working on something called CFF, or consistent force field, which was an attempt to simulate the properties of any molecular system based on potential energy. So after they were working there, Levitt went on to work on structural modeling at Cambridge, and Warshel continued to be interested in computer simulation of organic molecules. In 1969, Warshel did his postdoc in Karplus's lab at Harvard and expanded the CFF model into a program called CHARM, which stood for Chemistry at Harvard Molecular Mechanics. So the 3 scientists have been very long-time collaborators for almost 50 years, and it's I think interesting and I would say it helped me understand their work a little bit better, that they really began most of their work in physics and chemistry, as the Nobel Prize suggests, because there are some kind of formulas and references within the article that I kind of scratch my head about, because just based on my experience in molecular biology, they weren't things that I had heard of or within the field of biology. So this picture here is a it's a picture of the IBM 5100, which was introduced in 1975, which is the year this article was published. And it's billed as the world's first portable computer. So I just wanted to provide this as just kind of an illustration as to what the state of the art that this article was up against at the time it was written, and just remind everybody that this article was really developed, and at the time the research was being conducted on mainframes. So the problem, really, I think, as they defined it early on, is that the local structure, the sort of sequence of residues in their proximity, has not shown how residues distant along the chain can come together to form the overall conformation. That's how they characterize the research that had been done to date at protein modeling. So, I mean, what I took away from this, or as I kind of putting it in what I view as more layman's terms, which is that what we now call the primary structure, the amino acid sequence alone, it doesn't explain and isn't explaining how proteins behave. They're three-dimensional and they fold. So they came up with a fairly elegant solution as to how to how to provide better predictions and better modeling, and through averaging of the kind of key molecules within the amino acid chains. At the time, the approach had been really to try to keep track of every atom within the amino acid sequence, which was something that was impossible, particularly in light of the limited computing particularly in light of the limited computing power at the time. So they found that much of a protein's fine structure is eliminated by averaging, and the complexity in the chain can be lost with, or some of the needless complexity in the chain can be reduced without losing, a lot of the predictive modeling. So they use this averaging approach based on using a simplified structure where each residue within the amino acid chain is represented by two molecules, one being the alpha carbon atom and the centroid of the side chain within the amino acid residue. So this is an example of how the model pans out. This is from the Nobel Prize, the Nobel release and the summary of their work. This is somewhat similar to the figure 1 within the article, in which they provide a little bit more simplified stick drawing of what's going on here. So we move from the kind of complex individual atom mapping within the amino acid chain to averaging based on the centroid and the alpha carbon, and then that gives us kind of these clouds, this string of pearl like drawing that can be used or is used within the simulations. It's interesting also that the Nobel Committee had credited this research as reconciling classical physics and quantum physics within the field of biology, which is a pretty bold assertion. But I guess if you're going to give somebody the Nobel Prize, then certainly that's something that's worth doing it for. And this is just figure 4 from the article. So once you kind of have this average representation based on the two key molecules within a residue, then this can be used to create a model of how a protein actually folds and how the process works. They're a little light. The paper is a little light on details as to how they actually move from predicting the movement of these molecules at different within different conditions, and how they predict the movement of the key atoms from 3 steps in the folding. It talks about the folding of the idealized protein, and as the protein produced based on the averages, can be simulated by solving the equations of molecular dynamics at sufficiently small time intervals. So And the equations of motion can be approximated by Langevin equations where the charge of the variable is directed down the energy gradient with a random detection due to Brownian motion. Now, I think Brownian motion is a concept we're all familiar with, and molecular dynamics is a term that I've generally heard used to refer to this process of predicting protein folds. But the Langevin equations that they reference, that's not something that I can say I really have any understanding of all, at least. But they do provide us with an illustration of this process using, beginning with a different sets of randomized temperature and how the protein folds across this. And I should say in this particular article, they were using the bovine pancreatic trypsin inhibitor protein, and they use this because it's a relatively simple protein with only one amino chain, and so it was something that was easy for them to look at and resolve by X-ray. So to be able to kind of check their work, and also something that required relatively low computing power to predict. So why does this matter? Well, they kind of tell us why it matters towards the end of the article, which is that they started off kind of criticizing the attempt to understand everything that happens within the protein folding process, and the focus on every single residue and every atom within it, and suggested this fairly elegant averaging approach as a way to get kind of more predictive bang for the work that you do. And what they say is that as additional information can be gained about the protein fold, you can add additional levels of complexity to the averaging model, so that you get more detailed and more accurate folds and more accurate models of the fold as you gain more information about the protein. And then their conclusion is such a hierarchical approach might eventually lead to an understanding and simulation of very complicated biological processes, which has happened, as they predicted in this article nearly 40 years ago. So this is just an example of some of the the modern the kind of predictions and models that have come out of this research. This is what's in the background here. This image in the background is an image from a PyMol source from the protein data bank. So as if you take a closer look at it, you can see that there are actually coordinates for every single atom within the protein residue. And we have been able to develop very complicated models moving from these initial averaging based computer simulations. So these also give us this ribbon diagram down at the bottom is actually a JMOL representation of the same protein that you're seeing behind it. It's the E. coli 16S protein from the protein data bank. And off of the left, what we're looking at is just the PROSA program, which is used to evaluate different models of proteins. So, let's say, you have a protein that has not been resolved by X-ray or by NMR. It doesn't have its own, protein data bank record. You can use but you can find a similar protein. You can find the same protein in another species that has been resolved. You can now use these computer simulations and the data that we've gathered from them to use the resolved protein as a template to predict what your unresolved protein may look like. And you can use these tools such as PROSA to evaluate how accurate your model is likely to be. And whether or not the model that you've developed based on the similar protein falls within what's expected of proteins and reliability within that have been resolved by X-ray and NMR. And the other reason why this matters, and just kind of back to what I talked about at the outset, this is something that just kind of really got me interested in the field of bioinformatics in the first place. Which is when I read Rob Carlson's book, Biology is Technology. And in one of the chapters of the book, if any of you haven't read it, I'd highly recommend it. But in one of the chapters of the book, he compares the evolution, or I should say, he compares how biology has progressed and how biotechnology has progressed to the development of aviation, particularly from the 747 to the 777, but even in the early days of aviation. And he talks about how the thing that really allowed aviation to take off and for people to gain confidence in it, for it to gain wide acceptance, and really allowed an aviation boom, and for us to all, get on planes and fly every day without a second thought, is the development of accurate computer modeling that allowed aviators and engineers to predict how a plane would fly before they actually put it in the air, and to develop safer planes and accurate predictions of how planes would fly without actually crashing them. Carlson uses the same example of the importance of computer simulations in nuclear weapons in this quote, talking about how the United States, has continued to build nuclear weapons without actually having to conduct nuclear tests, just based on computer models. But in biology, we're not close to that point. And so that's where I just want to stop the slides and open up for discussion. And particularly as to, what is the role of computer simulations within biology, and are we ever going to get to the point that we can we can predict the way molecules, or the way biomolecules behave, the same way we can predict the way aviation does. Apparently, I bored you all to tears.
I'm curious, does anybody know what the state of the art is right now in terms of that ability to predict the shape?
For protein folding?
Yeah.
Yeah. People primarily use Rosetta now as just a pure prediction tool. So, and I think actually Rosetta is open. Trying to find out. I think you can download Rosetta or a version of it onto. It's free. I think it's free software. Anyway, people tend to use that. It's like an energy minimization software. I don't know much about it otherwise. To predict protein folding, and
so
there's also the Fold It challenge, and that was an extension of that. Yeah, more or less.
That's actually a question that I have, actually, because there was a recent announcement about Fold It, and there was a 10-year-old problem with HIV that was recently solved. I don't know if you guys heard that, which is in like 10 days or 2 weeks or something, which is interesting. That's not the first time it's happened with Fold It. How does Fold It? I would like to understand more about how Fold It works vis-a-vis these protein folding algorithms.
I played with Folded, and it doesn't actually have any algorithm. It's a human sitting in front of the screen and folding it manually. That's how it works. There were great stories about teenage kids folding proteins better than supercomputers and PhDs because they played with it like with their usual games, and they spent nights and days and weeks and months in front of it, and over time they developed some intuition that cannot be formalized, but that intuition is so good that they can fold it better than supercomputers.
I guess my question is, though, so I get the difference between human maths collaboration labor and computer software programs, but I guess what I'm driving, and I recognize that Fold is a manual process, what I'm trying to get at is, what is the difference? I'm really just trying to understand what the difference is between these programs like Rosetta, whatever that you mentioned, Nina, and Fold It in terms of, Fold It must have certain rules and about what can be done and what cannot be done. They're just not randomly folding these things together, right? So what's the difference of that and an algorithm?
And I think Foldit does, it has an algorithm that determines the free energy. It's just,
Yes.
it doesn't know how to, it doesn't walk through the entire space of the 200 or 2,000 variables. It lets the user kind of navigate through that space.
It's semi-automatical actually, because you can fold something with folded, and then if you see that the free energy goes off, like you move something and it's spiked, and so what you do, there is like a button, shake it, it shakes it a little bit, and goes down the energy, or just move it randomly, etc. But you can do everything manually, and every step you move, it calculates the energy and shows you the graph, and also when the chains of the proteins goes too close, it highlights them with red, and and marks it as impossible position, so that in reality they cannot sit in this place, because they will act on each other too hard, and it will repel those two parts of proteins. But that said, it's mostly a manual product, and from what I understand, the automatic things try to move it randomly and find this perfect solution. But because there are three dimensions and thousands of proteins, thousands of amino acids in a protein, there's space that you need to check. Mathematical space is so huge, and even the most powerful computers on the planet can do it. There is a project called Folded at Home, where they use distributed computing, and by now it's more powerful than the most powerful computer on the planet, and they're trying to fold proteins, and it's very slow. So,
I have a basic question. All of these algorithms, including Folded, which is human-based, and Rosetta, is the assumption here that the final three-dimensional shape, the final tertiary structure, going to be the lowest energy state, or is that the assumption? Because or is there plenty of examples? Because I know proteins obviously can undergo multiple conformations, and a lot of these active conformations are not in the lowest free energy state. So is there? Do these algorithms take that into consideration, or are we just going for the tertiary structure that has the lowest free energy state and assuming that is the structure just because it has the lowest free energy state? As you go down the energy well, there could be...
I don't know, to be honest. I know that they're looking for the lowest energy state, but I don't know if they take into account water or something like that.
Right, that would be the lowest global energy state. But, so like if you just draw like an energy well, there could be higher states, right? I don't know. If since we have all of these three-dimensional structures out there, I don't know this is, I legitimately have, curious about this. For all the structures that are already known to like really high, angstrom resolution, are there some structures out there that we have crystallized that are not in their lowest energy state, that are existing quite far from it, or are all of the crystallized structures at their nearest lowest free energy state?
I don't know. I could imagine scenarios where a protein, well, I guess depending on how it's crystallized, if it's crystallized bound to a ligand or another, protein or something along those lines, I could imagine it not being in its lowest energy state.
Right, but could you have one that fluctuates between, let's say, three conformations, and all of them are not in their lowest free energy state? So, like, so your like most stable state inside of a cell compartment, wherever it normally is it just happens to be at some local minimum, but not at global, right? Mm-hmm. And do these algorithms take that consideration, or is that the case for everything that's crystallized? Are there things out there that are known to kind of putter around, but always at some local minimum, but never at a that maybe the lowest minimum would not be the right conformation? I don't know.
Yeah.
That's just an assumption.
How would we measure whether a crystallized structure is in its lowest energy state?
I mean, I would think maybe just at that point you would have to go into theory, right? Like you would have to go into, whatever algorithms you're using. Like, I don't know. I mean, if you fed this, just for as a trivial example, if you fed this example into what's the one that everybody uses, the one, not Rosetta, or Rosetta, or the one where humans are moving it all around. You could, there's clearly an algorithm that will give you a number that says that you can reach an even lower energy state, right? And so could you take a crystal that's out there of confirmation and move it to an even lower energy state? And which one would be right at that point?
I haven't used Rosetta, so now I definitely feel like I'm missing something. I can, looking at the folded page, it says that I know they rely on free energy, but it also says they look for, the rules are, you're trying to get the protein as small as possible, so you're trying to get it compact. It's also based on hydrophobicity. You don't want any of the hydrophobic side chains to be in your water. And then you want to avoid steric collisions. The third one, it just describes as, clear the clashes.
And all those numbers are definitely going to drive up the energy. If you, obviously all of those numbers will, give you a value that is going to not put it in its lowest state. I'm just wondering if that's always needs to be the case.
They I don't know what it means, but they said looking for native state for protein.
Yeah, I guess I don't know either. I guess native state would be in some state that it's normally found in, whatever it is, that, maybe like not bound to a ligand in the cytoplasm under some physiologic condition would be native state.
Yeah.
With solvents at some concentration.
But even if we assume that they don't take account all the complexities, they still give you the first approximation of how it could look like, and then you can go and hunt after the full structure and having some predictions.
Yeah.
My impression, though, is so far our predictions really are not necessarily very close. I mean, even looking at the stereoscopic images they had in this paper, he claimed them to be close, but they don't look close to me.
Do you think we'll ever get to the point that we can rely on predictions of protein folding and of other biological systems at the molecular level the same way we rely on them, the same way we rely on predictions about how airplanes will behave without crashing?
That's the hope, right?
Yeah, I don't know, because it's not even just protein folding. It's this other level of being able to engineer binding pockets into proteins and, find ways to disrupt specific protein assemblies with small molecules, or, there's a whole cancer target therapeutic research going on right now to find small molecules that bind proteins that are, indicated in, different types of cancer. So it's just it's so funny. It just keeps on building and building. And everything has to be double checked on the bench. So, I don't know. I'm on the fence I'm so skeptical about any sort of prediction, but maybe that's just the bench scientist in me, where I feel like I need to double check everything, experimentally. But the hope is that we can get to that point. It's high throughput. I mean, that ultimately it's what it comes down to is finding ways that these high throughput methods of surveying a landscape, are possible. So.
And I think that comparing it with airplane model and even nuclear explosion model is not really correct comparison. I would compare it with weather modeling. Because airplane modeling, you got a liquid on that speed. It's a liquid, flying around and that's all you model. You got a liquid which is pretty consistent, and you got the fluid motion laws that are the same. It's one formula. You punch it in the model, you punch your structure of airplane, and that's it. You can go higher in resolution, you go better your model. The same I assume is explosion. On that speed is just liquid motion. Everything becomes a liquid. Whether the biology, you have billions, billions, trillions of different chemicals interacting in a random order with probabilities. It's more like a weather modeling to me. And in weather modeling, from what I understand, there is a known fact. There is a wall of prediction beyond which you can't go, because the errors they accumulate. Like interest rate, they keep adding up, and after some point, doesn't matter how precise you measure, how precise you model, the error will eat your model. So people who model weather, they don't even have any hope to be able to model weather beyond 2 weeks, because they say it's an impossible period. It's the same here. I think in biological system, there will be some model, but the precision will be always limited, and it doesn't matter how good our model, how much hardware we can throw in it, it will always be the average model,
I think that's true to an extent, right? I mean, it is just an analogy, and you're right that it's a very active system. So proteins have a turnover, right? So they will fall apart and get all floppy and get degraded, and then they get proteasomes come in and get them out of the picture. So I mean, all these crystals are snapshots, right? So, and different states. So it'll be accurate for whatever the half-life of that protein is, and then things will just get more disorganized over time. So it is sort of like an airplane in, I guess, turbulence, right? It's falling apart.
No, my point is different, that the structure of protein is not fixed. It's never fixed. What you get in the most probable structure, but you actually can't guarantee that it's even going to be that structure 99% of the time. It probably will be less. Most of the time it will be wobbling and having completely different structures. So even if you could go on, sorry.
I was just going to say, it depends on the protein, right? Some of them are incredibly rigid under very high temperatures, and depends on the portion of it. And some of them, like the loop regions that connect some of these, alpha helices, are very floppy and wobbly. So, it also depends which part of the protein you're looking at. So, there is that variation, yeah.
That's what I'm saying, and it means that with different probabilities, it will behave differently. It's like 0.1 probability will behave X, 0.5 probability behave Y, and 0.8 probability and behave Z. So, and then that's one protein, and then you got 10 proteins with probabilities. It's a mess. After some number of proteins you put in the picture, the margin of error becomes so big that you can't predict the whole system. It becomes like a weather, predicting weather 2 months ahead,
Hi there, folks. Sorry I am joining a little bit late here, but I was just, I wanted to chip in that the early part of my career, I was doing a lot of this simulation. So I just wanted to know if there were specific questions that you'd like to ask around some of the questions they had earlier.
We're trying to figure out how relevant these simulations are, these prediction programs versus vis-a-vis folded. For example, the question for me is, if I had some protein issue down the road, would I want to focus on trying to solve it using one of these protein prediction programs, or would I just put it up on Foldit and let the public solve it for me?
Well, so the original work on protein folding and optimization was done to be able to figure out what the structure was from X-ray crystals. when you take an X-ray crystal, an X-ray crystallogram of a protein crystal, that assumes that you're able to even get a crystal crystallized of the sample protein. But what they've done is that the X-ray has certain diffraction properties, and you wind up having to perform simulations just to be able to correlate what the expected protein structure would be with the various diffraction angles that are happening in X-ray when you're doing an X-ray of the crystal. So when you're doing that X-ray crystal, you're not sure. There's a process of interpretation and an intermediate calculation that are having to happen. Now with NMR, you can actually measure the distance between various residues. But again, that's an extremely complicated and mostly artistic endeavor that that you wind up having to do the same sort of thing, where you have some basic information, maybe a handful of dihedral angles and proximities, and then you have to go through a series of optimizations from that initial structure, where you're tying, the variables that to fixed settings, and then letting the molecular model sort of optimize around that. So the important point to take away from all of that is that this is not a science. This is very much an art. It has always been very much an art. And a local minimum doesn't mean that you've necessarily found the global minimum, or that the quote unquote global minimum in a vacuum without any water molecules around them is in fact the minimum. So just take everything that you're reading with a grain of salt here. Is the takeaway.
So, Matthew, what direction do you see this progressing then? Are we going to be able to improve the models where we can become more accurate, or is it going to go some other direction? Are we going to become less dependent on models?
So modeling is extremely important within a sort of constrained boundary where particular information or data that you're starting with, and then you're going to work from there. Doing it de novo, or designing proteins de novo, is never, it's computationally intractable. The analogy that was made earlier, that it's weather, is extremely is much art, interpretation, experience, and from my perspective, I don't want to be predicting the negative here, but because there's a long line of people behind me that have predicted negatives, and it's turned out otherwise. But that is useful. In other words, that's not an end. That it's typically used some other details that no. And my experience has been that in the last 20 years, there has been no movement in this field. We've maybe moved a couple percent closer to real structures, but a lot of it is still just we know that certain amino acids are more conducive to forming helixes or beta sheets or the like, and that gets you so close. Just knowing what the primary sequence is, that that there isn't a lot of actual improvement up and over that. And I'm not a big believer in fold it. I think that yes, you can work through a massive number of permutations, and you can get the feeling, the sort of gut feeling that you've gotten it correct. But is that something is going to, or have you just optimized it within a couple percent of a local minimum that's not actually relevant physiologically? So hate to be the bearer of bad news, but
So Matthew, do you think this is a problem of we don't have good enough models, or is this a problem of is this a biophysics problem that we don't yet understand?
So it's an interesting mathematical problem, right? Because the early models were you were referring to what is the Langevin equation, that there's definitely something to what we're looking at here. That's why it's a Nobel Prize winning sort of thing. Is that there's definitely a link between equations of force but when you start looking at extremely complicated molecules like proteins, it becomes more difficult to predict the tertiary structure, because you're talking about extremely shallow wells that if you just I guess imagine if you're trying to find a valley on a particular landscape. So and you're in the Alps. You can find a particular valley fairly easily because you look for the spaces between the mountains. But then imagine trying to find a valley in Nebraska where it's flat everywhere, and there are definitely changes in altitude, but you could miss a local sort of divot in the earth, and that's your actual valley that it's settling into. It becomes much more computationally intensive. It becomes much more uncertain when you're talking about finding valleys in an area like Nebraska versus finding an area valleys in an area like the Alps. And we don't even speak of it barely has hills. So yeah, that's the scope of the problem. Is that when doing either Monte Carlo simulations or some kind of random walk across a particular set of equations. It walks very slowly because there's lots and lots of little divots that it gets stuck in, and you get to make hops out of that. But if the bowl is particularly large, then if you're talking about a minimum that for instance, the free energy of two atoms that are say on opposite ends of the protein, as long as the molecule is completely stretched out, there's no repulsive energy between those far ends. But then if you turn an angle in the middle of the protein and bring those two ends closer together, at some point they're going to be attracting each other because there's a certain amount of electrostatic energy that's favorable. But then as soon as they get within touching distance or bonding distance, that energy goes up very, very quickly. And obviously there's a big electric repulsion between atoms that are within bonding distance. So yeah, it's not so much that the models are wrong. We're fairly confident we have good models and we have an accurate degree of expression, but it's time. You just don't the number of possible combinations valid are nearly infinite when you're talking about proteins that are longer than more than a dozen bases in length.
I would like to give another example explanation to that from a more mathematical standpoint, so probably that helps someone. If you imagine a formula that describes interaction between amino acid or atoms within the long protein, it's a mathematical formula where you can punch a position and type of every molecule, and it will give you an energy of the entire of every atom in the molecule, and it will give you the energy of the entire molecule. But unfortunately, that formula is such that you can punch in the energy and get the positions of the molecules back. It's unsolvable in an opposite direction. And the only way we can solve it right now is to try punching different positions into the formula and see if it approaches the energy level we are looking for. And there are some tricks that allows us to simplify the path, so we don't need to check all the possibilities, all the recombination of all the positions. But still, the amount of punching is infinite. And there is no solution to that formula. Nobody was able to come up with universal, way of taking the formula that describes a particular molecule and solve it, run it to the minimum. There is no such solution in mathematics. I guess a person who would come up with a way to find that minimum will be in one line with Newton and Einstein, probably above Einstein and above Newton, because that's a problem not only in molecular biology, it's a problem of entire physics, quantum physics everywhere. Electricity. There is a whole class of formulas that describe a lot of physical processes that are not solvable exactly the same way. It's not like a biology problem only. That's why with model is literally we're trying to randomly find the solution. Modeling is not like we're solving it. We just trying to find the right combination. And if you get lucky, model spills out something very fast. If you get unlucky, we stuck. It goes infinite. So that's how it works. And maybe you can correct me if I'm wrong, because I don't have much background in biology or chemistry, but that's my general understanding of the
No,
physics.
you're dead on. That it's interesting that you bring in Newton. It's the Newton method of solving of solving equations.
Yeah, Newton came up with a way of writing down the equation, so he got sorry. So Newton became
that the method that we're currently using, that's state of the art for doing a lot of these optimizations, was developed by Newton, right? And that's
no one get better after that.
no one's gotten that much better. There's a couple different tweaks, and you can start with different starting points, but yeah, by and large, it's Newton's method of minimization that is used to solve these equations for various minima, and that's how ancient this problem is, and how little we've come on this problem.
Yeah, and unfortunately, Newton only come up with a way to write it down and approximate it. He never came up with a way to solve it. And if I understand the state of the field in mathematics, there is a huge part of the mathematical science in the world that trying to solve this problem. It's not like biology works, and it's like a huge part of mathematics, huge part of physics. They work this problem day and night. People spend careers trying to crack it, because if somebody can even crack open it, it will change the world. So in a way bigger than anything else before. So that's how big this problem is.
So this is a little bit of a tangent from a discussion about simulation, but kind of a corollary would be, what is the state of the art right now for imaging the folding of a protein? Is it the X-ray crystallography, or are there other techniques? Is that going to get cheaper, quicker, faster, easier?
I think the majority of people to solve protein structures do X-ray crystallography. That maybe and some NMR, but I want to say mostly X-ray crystallography. And it is still quite a challenging process. I mean, just talking to researchers that I know in that community, there's always this like starting point on how to crystallize your protein, but then you have to make these slight modifications in order to actually form crystals of your protein. So they actually set up these very high throughput trays of their protein in hopes that one of those wells at some point in time will have crystals of their protein of interest. And one category of proteins that have proven to be extremely difficult to crystallize are membrane proteins. And if someone has more experience with that or wants to speak to that, please do, because I don't know if it's because of the mix of hydrophobic and hydrophilic amino acids, why it's difficult to crystallize, or it's difficult to pull from the membrane and form crystals from it. So there is a call out now, I think, an area of funding to come up with ways to better crystallize membrane proteins. I don't know if anyone wants to speak to that, but that's all I know.
How about so it's still difficult. Is the resolution improving?
It's not a matter of resolution. It's a matter of um, the the actual these signals sort of lay on top of one another, and without going into a lot of detail. And I was just noticing that that somebody at Annan's house is saying that my earlier statement about Newton minimization being the state of the art. Yeah, it's not the state of the art in terms of optimization, but the reality is the reality of the larger term picture is that in terms of incremental improvements and actual um, uh, effectiveness of solving these equations. Yes, there's in specific circumstances using other devised mechanisms for optimization, you can get better, or faster minimizing. And there have been, lots of different variants of Newton's method of optimizing things that have been developed, and it's usually Newton's dash something something something, with all the names of all the people that worked on the particular method. But when you start talking about NMR, you're thinking of NMR, or sort of MRI type pictures where it just looks like an X-ray picture, or you're actually seeing slices of a particular structure, but what you're looking at there is just different densities of water, and the water protons, the protons in the water. If you perform NMR, what you're doing is you're actually stimulating the nuclear spin of the nucleus of the proton in the nucleus of the hydrogen atom, such that all of the spins are becoming parallel. They're all in a stimulated state. And the normal state of those protons is a mix of up and down spins. And when I'm saying up and down, that's just a way of thinking about it. It's not, up or down in the z-axis sense. It's up or down in terms of quantum states. There's it's up or down relative to wherever the magnetic north and south fields are. So uh, when you're doing a technique like NMR, you then are measuring how those nuclei are relaxing over time, and depending upon the other protons in that environment, how closely they're packed together, their ability to relax to a statistical mix of up and down versus being fixed as all up is going to change over time. And it's not like you can pick out, this proton is performing, is relaxing like this. It's a series of correlations. You're actually for some of the um, it's not ordered in sort of an X and Y axis picture. It's more all of the aromatic protons, all of the protons that are attached to phenylalanine, all of the sort of six-membered ring protons are at one side of the field, and then all of the aliphatic protons are at another side, and then the ones that are close to a carbonyl group, or the actual near to the amino backbone, they're all going to be clustered in another area. So you can look at the various correlations between those different protons, but it's not exactly it's not like a microscope where I'm going to finer and finer levels of visibility into that microscope, and then eventually I reach some perfect resolution with an electron microscope that's just going to let me see exactly what it looks like. These techniques are much more abstract and are measuring things in a different view that doesn't really correspond to what you would think of as like a microscope view. An NMR is a very different thing than a microscope.
So let me try to explain in other words, and you can correct me if I'm off in my explanation. But it's like making X-rays from all the sides and then merging them in one picture. And then what you need to do, you need to unmerge that picture. And you got multiple merges, so you got like 1,000 X-rays, and then they merge in different combinations, and you got those merges, and then you need to solve that riddle and unwind it. At least that's from what I understand what the crystallography is about.
Yeah, it's kind of like the lady in a box, right? With the magic trick where they stick the swords in, right? You're trying to figure out what the lady looks like, and you're sticking swords into the side of this box, and basically, when she yells, oh yeah, that's where she is. But literally, that's that's a lot like what you're doing with X-ray crystallography, except that the lady in the box is this crystallized molecule, and there are multiple repeats of the same protein structure. In other words, it's not a single molecule that you're visualizing.
There are multiple ladies with spaces between them.
Right, but they're all twin. They're all triplets. They're n many women that are all packed together, and they fit together like an Escher picture. That they're all nicely packed together. And when you beam an X-ray in at a particular angle, the combined effect of many of these women is that it bends at a 17-degree angle, and you're somehow trying to reconstruct. Oh, if I have 20 women lined up in the box this way, then this particular then the beam will be refracted off of each of them additively into a 17-degree angle, and that's
so in a nutshell, you're not looking through the microscope on the picture,
right. And so it's not when you start talking about things like resolution, it's not exactly a meaningful analogy. I mean, yes, you can use finer and finer needle-like swords, but you're still sticking swords into that box to figure out where she is. And obviously with NMR, you're going to get a different response for different types of molecular entities. So it's like you're sticking the knife in and you hit bone, right? Okay, there's a bone here, and depending upon how hard it is to stick that knife through, it's either bone or not bone, and you have multiple bones that are I could take this analogy to its horrific end, but hopefully that gives you some sort of way to visualize why this is different than a microscope.
Cool. Thank
I
you. Oh,
One more question. So while we were talking about the sticking stuff, so I get the question because it sounds more like DNA sequencing, what's going on there, and I wonder if somebody heard or read about more advanced of way of sequencing the structure, because crystallography or MRI, it's kind of a sequencing of a structure. It's more close to sequencing than microscopy. So we're trying to apply some process repeatedly and infer the structure from the output. I think there is more hope in that direction, being able to restore the structure of protein and try just to model that thing. And I remember vaguely a couple of weeks ago on a Nature podcast, they said that somebody came up with a way to put the proteins that are not crystallizable into the crystal and then do the crystallography. So sounds a lot of people work in the direction of improving the ways of, reading the structure rather than just trying to model it. So.
Out of curiosity, how much visualization of these structures is possible via atomic force microscopy? Is that a thing?
I don't know.
I would say it's probably not a thing. What we're talking about, I mean, atomic force microscopy is great if you can get a flat layer of thing that you're visualizing. With with modeling like the external surface of a crystal or something that you can just take a flat plane, a two-dimensional structure. But atomic force microscopy is going to be useless when you're talking about a protein, because you're more interested in what the internal cavity of the protein is. So that with a very large protein, you may have multiple dimers or tetramers or multiple copies of the protein that all take part of that conformation, and you're as interested in as what's going on inside that as you would be on the outside. And if you look at certain AFM images or even electron microscope images, what you're looking at is like the surface of the bug's eye. You'll typically coat the entire slide with metal. You'll do a either gold or aluminum deposition, because you can't actually get good electron diffraction from biological molecules. So what happens is that you have to coat the outside with a thin layer of metal atoms, and that's what you're actually visualizing. So atomic force microscopy, might be able to get you to the outer, glob, but it wouldn't be able to actually get in between all of the different crevices inside the molecule and be able to manipulate it the way you would on a dissecting microscope.
all right. From this conversation, I realized there is a lot I need to learn about protein folding. I don't know if anyone has any final questions to ask, but I would like to thank Dan for leading this discussion. And it's always nice to talk about something that I know absolutely nothing about because I learn the most in those situations. So yeah, I really appreciate it. Thank you.
Thanks, Dan.
Thank you.
And maybe at some point in the future, we could follow this up with a protein crystallization, like a current protein crystallization paper or something along those lines, or techniques that people are using to crystallize proteins and get a good handle of what's actually happening in the lab as well. So that might be a good follow-up to this.
Yeah, I'd be interested in that. That sounds good.
Yeah, I think that would be cool.
It would be cool in general to talk to someone who works in the field of protein folding, who can just answer our questions about what they use, what works, what doesn't work, and just answer the question we just had. But from so much alive, so he knows the state of the art, she or he.
Yeah, absolutely. Yeah, we should put our feelers out there and see if any of us happens to know someone.
By the way, it's a great idea for a journal club to invite a speaker who's familiar with the field and interrogate him for 2-3 hours.
Yeah, absolutely. I agree. And that's a perfect transition to what I want to talk about. Yeah, I want to